to our channel. This is Science Lectures 256. Don't forget to like, subscribe, and follow us on our social media handles. So we are still continuing with our Mendelian genetics, and we are looking at those extensions that Mendel did not encounter. We said one of the reasons why he was successful, he never encountered any gene interactions. What are gene interactions, by the way? Gene interactions are conditions. These are conditions where genes act to modify a phenotype. Where genes act to modify a phenotype. Those are gene interactions. So in gene interactions, we have one or more genes working together to give a specified phenotype. And there are different types of gene interactions. They can be allelic gene interactions. You can have what they call non-allelic gene interactions. You can have what they call intra-allelic gene interactions. You can have what they call inter-allelic gene interactions. Inter-allelic gene interactions. Allelic gene interactions is where we have genes. These are interactions between genes located on the same locus. These are interactions. So we say allelic gene interactions are interactions between genes located on the same chromosome. The interactions between genes located on the same, homologous chromosome. Same homologous chromosomes. Then you have already guessed what non-allelic gene interactions are. Non-allelic gene interactions. These are gene interactions. All these are interactions between genes located on different chromosomes. So those are allelic gene interactions and non-allelic gene interactions. Allelic gene interactions are synonymous with what they call intra-allelic gene interactions. Intra-allelic gene interactions. So what are intra-allelic gene interactions? These are definitely gene interactions. These are gene interactions or allelic interactions. Or you can say they are interactions between alleles of the same gene, located on homologous chromosomes. Homologous chromosomes are chromosomes that have similar genes. I want us to look at some of the examples of intra-allelic gene interactions. The first one, we looked at it, which is complete dominance. And we said Mendel in all his experiments encountered what they call complete dominance. The second one is codominance. We looked at the examples of various under codominance, the coat color in cattle. We looked at the AB blood group system in humans and the sickle cell anemia trait under codominance. Then there is what they call incomplete dominance. In incomplete dominance, we say the phenotype of the offspring is different from that of the parent. Then, and we looked at the color in snapdragon flowers. Then there is what they call super dominance. dominance. When you read some text, they will call it over dominance. Over dominance. So let's look at this briefly. Superdominance or overdominance is a condition where the offspring phenotype is superior. If it is not superior, they can say it is above the phenotypic range. Of homozygotes. So superdominance is a condition where the offspring phenotype is superior to both parents, or where the phenotypic range of the offspring is superior, is above that of homozygotes. And I want us to look at some of the examples under the superdominance. The first one is sickle cell anemia. When we are discussing codominance, we talked about what they call heterozygous advantage. So you realize that individuals having that are heterozygous for sickle cell anemia, especially in the tropics, have what they call the heterozygous advantage, and their chances of being infested by the malaria parasite are low because of the results that we gave. The second one is the salmonoid major histocompatibility genes. Histocompatibility complex genes, or what they usually summarize as MHC genes. So these genes are responsible for coding for certain peptides, proteins on the cell surface membrane that are responsible for that play a role in the immune system. So when scientists use a specific type of fish under the salmonoid family, that is the Arctic carp, they discovered that the type of fish that had heterozygous genes for the MHC, meaning that, for example, if I have MHC1 genes and MHC2 genes, when they got fish that had the MHC1 and others that had the MHC2 genes and they fertilized them, they obtained an individual that had both MHC1 and MHC2 genes. But we are looking at superdominance. We said that the phenotypic range of the offspring is above that of both homozygotes. So those Arctic fish that were heterozygous for both MHC genes had few cysts of parasites, because these fish are usually affected by monogenians, which usually tend to form cysts in the various body parts. But these fish that were heterozygous for both MHC genes had few cysts, and they had better chances of survival. And actually they tended to produce more offsprings. Better chances of survival even when the temperatures went on alter, netting their survival mechanisms were high compared to the homozygotes. The last example that I want us to look at is what they call the black vanilla orchids, or what they call Gynadenia relicana. Gynadenia relicana. If they don't call it Gynadenia relicana, they usually call it the black vanilla orchids. So This is a plant that is very common in the Alps, and scientists wanted to establish, when they sequenced its genotype, they discovered that it had genes that code for anthocyanin, and these genes were called the GYMNRB genes. And these genes were located on the 612 and 632 position on their chromosomes. And these two genes coded for some amino acids that were responsible for the formation of anthocyanin. So, how does this bring us to superdominance? Initially, the plants that used to exist had the black and white alleles. The black and white alleles. But they discovered later that the population of the black plants was decreasing, and the population of the white plants was decreasing. And there was a new phenotype that had emerged. That was the red variety of the black orchid, or the Gynadenia redicana, red variety. And this red variety, its population was rapidly increasing in the Alps. And when they tried to check its genotype, they discovered that it had both black and white alleles, which made it a heterozygote. And they discovered why was it able to survive. It attracted a lot of pollinators. Ranging from bees, spesam flies, a lot of members of allelic, Doptera, which are the butterflies, and the moths, which meant that it produced a lot of seeds which were easily dispersed. And as of today, the red variety, which is heterozygous for the both and black and white allele population, is very high compared to individuals that have only the black or white allele, which brings us back to superdominance over dominance. That the offspring is heterozygous and it's superior. It has a superior trait compared to both homozygotes. So that is it for intra-allelic gene interactions. We said intra-allelic gene interactions are interactions of alleles of the same gene located on the same chromosomes. And said it can be complete dominance as experienced by Mendel. It can be codominance or incomplete dominance or superdominance. So I want us to look at next, that is inter-allelic gene interactions. Inter-allelic gene interactions. And that is where we are going to spend most of our time today. So inter-allelic gene interactions, as you hear, the interactions of genes located on different homologous or non-homologous chromosomes. And there are many. We have what they call polygenic inheritance. We have what they call complementation. We have what they call epistasis. Then there is what they call redundancy, or what they call duplicating genes. All these are deviations from the normal Mendelian expected ratio. And as we're discussing the hybrid cross of Mendelian, the ratio was 9:3:1. But as we are going to see in all these, this ratio is not achieved. It deviates from that ratio. And we say inter-allelic gene interactions, but we need to see how do they work. Let's assume a hypothetical pathway of inter-allelic gene interaction. Interallelic gene interactions. So remember, we are dealing with genes. That means genes will code for a specific protein. Which protein I'm going to call molecule A. Molecule A. But this molecule A needs to be converted to molecule B. Molecule B. Then this molecule B will still need to be converted to the final product, which I'm going to call molecule C. Molecule C. But the conversion does not happen out of the blue. We need to have specified enzymes. And for the conversion of molecule A to molecule B is by enzyme 1. And for me to convert molecule B to molecule C is by enzyme 2. If I remove my enzyme 1, the conversion of molecule A to molecule B can never happen. And if I remove my enzyme 2, assuming I have my enzyme 1 here, the conversion of molecule B to molecule C can never happen. So we are looking at interallelic. All the genes that code for these different molecules are located on different chromosomes, but they work together, which is what we call gene interaction. I know this has not yet made sense, but as we are going to continue our discussions, you will see how this hypothetical pathway is used to explain interallelic gene interactions. So we begin with polygenic inheritance. Polygenic inheritance is where you have a phenotype, which is as a result of a cumulative effects of many genes. Cumulative effect of very many genes. For example, examples of this polygenic inheritance, things like weight, things like intelligence, things like height, things like skin color, things like eye color. All these are the result of very many genes working together to give us a specified phenotype. And you will agree with me that these things usually show little difference. If I try to plot a graph, let's say of height, the graph shall go on increasing, then it shall go on decreasing. For me to say that you are dark skinned, somebody is light skinned, I need to have two contrasting individuals. So it is very difficult for you to tell these traits. Actually, in genetics, you say that these traits do not have the secret units. Like you can't measure that this body's skin color is 1. No. You can only tell their presence after comparison over a large population. And the role of polygenic inheritance, it maintains what they call trait stability. In a population. Maintains what they call trait stability in a population. That is polygenic inheritance, where a phenotype is a result of cumulative effects of very many genes. In summary, very many genes code for one phenotype. Next is complementary genes, or what is called complementation. Complementary genes. So what are complementary genes? Complementary genes or complementation is a form of gene interaction where a phenotype is coded by two dominant genes of different allelomorphic forms, of different allelomorphic forms, giving a ratio of 9 to 7. So in complementation or complementary genes, we need to have two dominant alleles to obtain a specified phenotype. Having any of the alleles being not dominant or having any of the genes recessive gives us a different phenotype. What do I mean? So, some time back, scientists tried to carry out a certain experiment, and they got two white flowers, and when they crossed them, they got purple flowers. What I mean is that they got pollen from one flower, transferred it to another, and also got pollen from one flower and transferred it to another. And when they got the seeds, they planted them, and the product, this time they got purple flowers. But when they did the same mechanism on the purple flowers, they obtained some purple flowers and other white flowers. Then they tried to explain the genetics, which is what we are going to do today. So let's do a simple cross. Say in our process you begin with the parental phenotype. Say they got white flowers. Then we say you write the parental genotype. So when they tried to examine the genotype, they discovered that one had two dominant alleles and the other had recessive alleles. Then we say that these usually undergo meiosis. And you get AB. I'm supposed to get 4, but I will use 2 because they are all the same. AB, then AB. Then these will undergo, these are our gametes. Then this will undergo random fertilization. So this is A, capital B, small b, capital A, small b, capital A, small a, capital B, small b, capital A, small a, capital B, small b. Say this is random fertilization. This is our F1 generation. And we say there F1 generation, then our F1 phenotype is that they got all purple. So I hope you are following. I've said they got two white flowers, and when they crossed them, they discovered that they would yield purple flowers. Let's see what happened when they crossed purple flowers this time, and how does the genetics of complementation come in. So we said our parental phenotype is, now we are dealing with purple flowers, and another purple flower. What is the parental genotype? We have already seen from our F1 generation that it is capital A, small a, capital B, small b. Still I'm also having here capital A, small a, capital B, small b. When you say this one will undergo meiosis, and we are supposed to obtain 4 gametes. We said from Mendel's second law that separation of these genes is just random. So I can have capital A, capital B. I can also have capital A, small b, small a, capital B. Then I can have small a, small B. Then I also come here. The same. I can have this capital A coming with this small capital B. Then this small capital A coming with this small b. Then this small a coming with this capital B. Then this small a coming with this small b. So now since I'm dealing with 4 different genes, I would advise you to use a parity square. Earlier we discussed how do they use a parity square in genetics. So I come here, this is a woman and a man. I come and start filling in my genotypes. This one, I put it here, capital A, capital B. Then capital A, small b. Then small a, capital B. Then small a, small b. I'll put these ones on this side. Now I'm going to also fill in these gametes here. Capital A, capital B. Capital A, small b. Small a, capital B. Then small a, small b. Now we start doing the process together. I hope I'm doing as you are also doing. So this A comes with this A. So I have capital A, then capital B, capital B. Then this capital A goes with this capital A. Then I have capital B, small b. Then I have capital A, small a, then capital B, capital B. The same story, capital A, capital B, small b. Next is this one goes with this one. Remember, this one goes with this one will go with this one will go with this one will go with that one. Those are the rules we say that are followed when you're using a parity square. Then this is capital A, capital B, small b. Then capital A, small b, small b. Then capital A, small a, capital B, small b. Capital A, small b, small b. The same thing here. I will continue doing the same. We are also doing everything together. I'm not leaving anybody out. Yeah. I'm trying to complete this parity square. I hope you're also trying to complete yours. Yeah, I have finished the parity square. So this is where we have our random fertilization. Now write our F2 phenotypic ratio. Phenotypic ratio. So we say the phenotypic ratio is the number of expected phenotypes from a cross. Remember, we are dealing with complementary genes, and we say in complementary genes we are supposed to have 2 dominant genes obtain a specific character. You see that here I'm having purple flowers because I have capital A and capital B. Capital A and capital B. That means that having any of these genes recessive gives me a different phenotype. So you can see that me having the purple flower, I must have capital A and capital B. Meaning that whenever I have this capital A recessive, I can't have the purple flowers. Whenever I have this B recessive, I can't have the purple flowers. So from that simple explanation, I want us to deduce the phenotype together. We've said for you to have purple flowers, you're supposed to have capital A and capital B. So we are going to cross together and identify which flowers are purple. So this one is purple. This one is also purple. You can see the capital A and the capital B. This one is also purple. You can see I have capital A and capital B. This one is also purple because I have capital A and capital B. Then this one is also purple because I have capital A and capital B. Then now this one. So I have capital A, but I have recessive B. That is not a purple flower. Then this one is also purple because I have capital A and capital B. The same here. I have capital A, but I have B recessive. That is also not a purple flower. Then we are here. I have capital A and capital B. This one is recessive. Then this is purple. Then I have capital A, capital B. This one is also purple. Now I have small a, capital B. That is not a purple flower. I have small a, capital B. That is not also a purple flower. Then we are here. I have capital A, capital B. This is also purple. Then this one is also not a purple flower because I have B recessive, here I have A recessive, and here I have both recessive. So if I am to count, I have 1, 2, 3, 4, 5, 6, 7, 8, 9. Those are 9 purple flowers, 9 purple flowers, 2. The rest are 1, 2, 3, 4, 5, 6, 7. 7 white flowers. 7 white flowers. Remember we said 2 dominant genes of different allelomorphic forms giving a ratio of 9 to 7. You see now the 9 to 7 has come out. That whenever I have 2 dominant genes, I obtain the purple flowers. Whenever I have any of the genes recessive, I obtain a different phenotype. But one thing you need to know that we are dealing with complementary genes. Complementary genes. Meaning that they complement one another. This is like having a child. You need both a man and a woman to obtain a child. Having any of the sexes absent, you only remain with that one sex. If I am a man, I will remain a man. If somebody X is a woman, she will only remain a woman. She can never have a child. So that is how also these genes operate. That let's say gene A is responsible for formation of the purple color. The purple color. Then this gene B is responsible for the deposition of the color. Position of the color. Position of the color. So we have our purple color. Then this color shall undergo some modifications. Then it will be maybe deposited in the petals. Deposited in the petals. In the petals. By gene A, then by gene B. So having mean minus this the deposition can never happen minus the purple color. And also the purple color can be that sometimes it won't be deposited. So these genes work together so that you can have the purple phenotype expressed within the offspring. Having any of the genes recessive gives you something completely different. And we say the normal Mendelian ratio is, let's write it here, 9 to 3 to 3 to 1. But here in complementary genes we have 9 to 7, which is a deviation from the normal Mendelian dihybrid cross. Yeah, that is all about complementary genes. That is it all about for complementary genes. And next we are going to look at redundancy. Redundancy. Or when you read some texts, as I said earlier, they will be called duplicate genes. Duplicate genes. Duplicate genes. Duplicate genes. Redundancy, or what they call duplicate genes. So in redundancy, what they call duplicate genes? We only need one dominant allele. Dominant allele is enough or is required to have a given phenotype. Have a given phenotype. Have a given phenotype, or one dominant allele is enough to have expression of a given trait, and the ratio for redundant or duplicate genes is 15 to 1. 15 to 1. Let's use an example. Scientists try to use what they call snapdragons. So they got red snapdragons, and cross them with white snapdragons, and red was dominant over white. When they cross the two, they obtain red snapdragons. Now, when they recross these red snapdragons, they obtain some red snapdragons and obtain one white snapdragon. Let's try to explain this as genetics. Let's do a simple cross. Say our parental phenotype is a red snapdragon crossed with a white snapdragon. Let me first clear this. Then our parental genotype is, I'm going to use capital R. Remember, we are dealing with two genes, and I can use maybe capital W. Then this is small r, small w. So now we say this one will undergo meiosis, and they will yield for different genetic combinations. But I said since they are all the same, I'm going to use only two: RW, capital R, capital W. So I get small r, small w, small r, small w. Now these ones will undergo, these are our gametes, and these ones will undergo random fertilization, and I get capital R, capital W, small w. The same story here, capital R, capital W, small w. I won't complete them because they're all the same. And this is my F1 of springs. And so they were all red snapdragons. Now, when they got these two and they tried to cross them, let's see what happened. So our parental phenotype is now red snapdragons crossed with another red snapdragons. The parental genotype is now capital R, capital W, small w, crossed with another capital R, capital W, small w. So these ones will undergo meiosis to give us 4 different genetic combinations from Mendel's second law of inheritance. So after that, we said we shall load our data on a Punnett square. So capital B, small e, capital E, capital B, small b, capital E, then this and this. Okay? Wow. Now this, I hope you're appreciating a bit of genetics, a lot of diversity. Okay, then I start doing my process. This one with this one. Okay, this one, and this one, and this one, and that. Capital B, small b, capital E, small e. Then capital B, capital E, small e, capital B, small e, capital B, small b, capital E, small e. Capital B, small b, E and E. Same story here. Then here. Then let's organize these things here. Okay. Then trying to complete this Punnett square. I hope you're also completing yours. Yeah. Small b, capital E, small e. Capital B, small b, capital E. I mean small e, small e. Then capital B, small b, capital E, small e. Yeah. So we say, from what you can see here, for us to have the black phenotype expressed, we must have both gene B and gene E dominant. So as we are going to deduce the phenotypes in this Punnett square, we need to look out for those ones that have capital B and capital E. And let's start. So this is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, this is 8, and this is 9. So I have 9 black dogs. 9 black dogs. Then I have a condition where this E is recessive. This E is recessive. 1, 2, and 3, and 4. Have 4 when there is a genotype. Then I have also a condition where the B is recessive. This is 1, this is 2, and this is 3. So I have 3, have 4, I have 3 brown ones, 3 brown dogs, and 4 golden dogs. 4 golden dogs. So how are you asking? You're probably asking yourself, how did the brown come in, first of all? Then how did the golden also come to 4? Yet originally we had 1 brown dog. Okay. So in recessive epistasis, as you are in recessive epistasis, a recessive gene will mask the expression of another gene, therefore it will prevent the expression of that gene. From what we've so far discussed, you've seen that for you to have a black dog, you must have gene B and gene E dominant. So hypothetically, we say that gene B is responsible for the production of the black pigment. But for the expression of this black pigment in a dog, you need to have gene E. Gene E is responsible for the expression of this black pigment. So whenever I have any of these recessive, it will mask the expression of either this pigment. What do I mean? I'm going to use an example of this one here. This B, E, E. And I also use B, E, E. We say B, E gives us gold. Why? We shall have the black pigment being synthesized, but this pigment will never be expressed. So that is what it means to have recessive epistasis. This recessive E has masked the expression of this dominant gene B. At the same time, we can have this gene B recessive and this E dominant. However, we shall have no pigment being produced, but you have a gene that is staying in the skin, the hair shaft express a given color. That is how we end up with a browny color being expressed in the phenotype. So that takes us back to our hypothetical pathway, that gene B is responsible for pigment production, and actually we assume it will also produce enzymes that will be responsible to see that this pigment can undergo various modifications. Then this gene E will produce enzymes that are responsible for the expression of this black manufactured pigment in the phenotype. Having any mutations in either gene B or gene E will give us completely a different phenotype. And the ratio is 9 to 3 to 4. Hope that is understood. So let's sum up everything with a very simple table that summarizes whatever we discussed today. So we said, let us write the interaction. We draw a simple table here of interaction and the ratios and the ratios and the ratios. The first interaction, we see the normal Mendelian gene interaction is what we call the additive gene interaction. Additive gene interaction. And say the ratio is 9 to 3 to 1. After that, we started looking at the various deviations from this ratio. The first one was recessive. We looked at recessive epistasis, which you've just seen. That one, and we say the ratio is 9 to 3 to 4. We looked at dominant epistasis. Epistasis, and the ratio we say is 12 to 3 to 1. We've looked at also what they call duplicating genes, or what they call redundancy. We say the ratio is 15 to 1. And you can see all these three deviate from the normal Mendelian ratio, which is 9 to 3 to 1. So in this video, we've discussed the various forms of gene interaction. We say they can be intra-allelic or inter-allelic, and we've seen the various inter-allelic interactions. We also saw the intra-allelic ones. Till next time.